infectious diseases, research, medicine, health. Welcome to Outbreak News Interviews. And now, broadcasting from the Outbreak News Skylar Studios in beautiful West Central Florida, here is your host, microbiologist and editor of OutbreakNewsToday.com, Robert Harriman.
Well, hey everybody, this is Robert, and welcome to Outbreak News Interviews. Now, earlier this week, scientists with the Scripps Research Institute announced the discovery of antibodies in the blood of recovered COVID-19 patients that provide powerful protection against the SARS-CoV-2 in small animals. The study was published this week in the journal Science. Now, here to discuss the research is Elise Landais, PhD. Dr. Landais is a principal scientist with the IAVI Neutralizing Antibody Center at the Scripps Research Institute. Dr. Landais, welcome to the show, ma'am.
Thank you, Robert. I'm happy to be there. Thanks for the invitation.
Before we get into the research, I just want to get an explanation for the audience that may not be aware. What is a neutralizing antibody, and what's its role in the treatment of disease?
So antibodies are rather large molecules that are generated by our white blood cells in response to infection by a pathogen. They typically have this Y shape with a body and two arms. And then each arm can specifically recognize 4 pieces of a given pathogen. And then once bound to their specific target, they can have several effects. One is as they directly bind to the surface of a virus or bacteria, for example, they can physically block them from infecting your cells. And that is what we call neutralizing. So it's physically putting a barrier between the virus and your cells. So then it cannot infect. So it's neutralizing infection. The second effect, it's also because these antibodies can recruit other molecules and cells of the immune system, and they can also promote destruction and elimination of pathogens themselves, but also of infected cells. And through this blocking and neutralizing antibody function, they can prevent infection in the first place, obviously, but that is what we're trying to achieve with the vaccine. And then basically we're trying to teach your body to make these antibodies before being exposed. But then once you have these antibodies floating around, if you're exposed to the virus, they cannot infect you. But as the vaccine is not always available, like it is the case for this COVID-19 disease, these antibodies could also be injected to provide protection, as we've already isolated some good ones from other people. Okay. But then can also be used to treat and promote elimination of already infected cells, and then help people who are already infected through the neutralizing activity, also to prevent reinfection of new cells and then improve disease progression.
So it could be used like a immunoglobulin too, like preformed antibody injected into somebody?
Correct.
Yeah. Just like for rabies, for example.
Yes.
Now, this development, is this something that you've done with other infectious diseases, this neutralizing antibodies?
So there's a long history of using antibodies to treat and prevent disease before there were antibiotics over the treatments, such as vaccines. So for diphtheria, tetanus, and polio, for example, they are all they're already licensed antibodies to prevent respiratory infection, for example, in premature babies. And the rapid isolation of these self-neutralizing antibodies was really made possible by leveraging the technologies and expertise we already have built over the past decade for HIV also, and we are also trying to use this technology for HIV as well, where we have isolated many powerful neutralizing antibodies that are currently being developed eventually for protection and cure, and could serve as a template also for vaccine design.
Well, let's go ahead and talk about the research and the process. And I looked at the study, and it looks like quite a lengthy process. What did it take for you and your colleagues to do to isolate these antibodies?
So you're right that it is typically a lengthy process. So this is something that typically takes a year and a half from the moment you decide that you're going to do the project to animal studies like we did. So really, first and foremost, what it took to get this done in less than 8 weeks was really an enormous collaboration, not only from individual scientists, but also from institutions. You mentioned Scripps and IAVI, but it was also done in collaboration with University of California, San Diego, with our colleagues there. So it was really three big institutions here, and then many scientists from this institution coming together. None of us could have done it alone, really. Each of us, we came with our own expertise, and then we all work in parallel to execute part of this work. So to elaborate a little bit on the process itself, so the first thing you need to do is access patient samples. And then once we have the white blood cells from these individuals, we need to go fishing for the white cells that are generating the antibodies that are specific for COVID. So we have special molecular tools for that. So we fish those B cells, and then from those white blood cells, we clone the genes that are coding for the antibodies that they are generating, and then we are able then to produce those antibodies ourselves in the lab, and then we can evaluate those antibodies for the functions, either for neutralization or for binding or other functions that we mentioned earlier. So that once we have those antibodies and we find some that are really, really good, then we can inject them in an animal model that we as either prevention or treatment. So in this case, we use the hamster model that is widely used for SARS infection. But what I want to add also is that beyond the technical work itself, it also took extreme dedication from everyone involved. It was very long days and short weekends, if any, and very little sleep over this 8 weeks. But it was really exciting. This is really what we're all meant to do, you know. As a scientist, it was our calling, right? So we were all just so very happy to be part of this, and we're very proud.
Well, congratulations to you all. Now, I know there hasn't been any work with humans yet, but what do we know concerning the efficacy and the safety thus far?
The short answer, I guess, is nothing yet, really, because what we've done so far is only a very small, limited animal study, and there's much more that needs to happen before it really moves to the clinic and evaluation in people. So safety and efficacy will be, of course, paramount and will be rigorously assessed at that point.
Right. And this is something that can be mass-produced at a low cost?
Yes, so typically antibody can be mass-produced, but it is typically costly. And there are some technical challenges to large-scale production, but then those are things that have been done in the past, like I mentioned. So we have learned from those examples before, and then we're currently addressing these issues for those antibodies specifically, and then we're going to move even faster now that we have the examples of the previous diseases.
Yeah. Okay, and final question. Dr. Landais, what's next? What's the next step?
So what's next is exactly that. Your previous question was just right on spot. Basically, we are working hard now to developing these antibodies to be able to ensure safety and efficacy. But also we are still trying to isolate even better ones if possible. And then IAVI is definitely committed in making these antibodies affordable and accessible globally, including low and middle income countries. While we're all waiting for a vaccine, we're hoping that this could be a countermeasure to at least to healthcare workers and then more vulnerable people. That's really our goal. However we can help, we want to move forward as fast as possible.
Well, very good. A very interesting study, and I'll link to it link to the study in the show notes when I publish the podcast. And I want to thank you, Dr. Elise Landais, for sharing your time and your expertise. I appreciate it very much.
We appreciate the invitation to share our work. Thank you.
All right. Have a great day.